Welcome, and thank you for joining us for our live virtual MDA Engage Nevada seminar. While we would have loved to have been in person for this event, we appreciate you all joining us virtually today. My name is Nicole Petrowski, and I'm the Community Education Specialist for MDA, and we are so glad to have you join us today for this important educational seminar. MDA is committed to community education. We believe in the power of bringing our community together for opportunities to learn from specialists and having opportunities to connect with others. This event is part of the larger MDA Engage series with disease-specific symposia, community education seminars, and community webinars as well. Our community education seminars, which we are having here today, are geared towards general neuromuscular disease topics such as best practices in care, research, clinical trial information, etc. While each event will feature regional specialists covering these mentioned topics, each event will also feature a specialty topic, which you can view on the screen. These events are events that we have lined up for the remainder of 2020. Our Engage disease-specific symposiums are geared towards a particular diagnosis, and topics include research, genetics, and others that are important to those diagnoses. To view the agendas for these seminars and symposiums, as well as to register, you can visit the MDA Engage section on mda.org. You will also find our past recorded events here as well for your viewing option. We would not be able to put on educational programming without the support of our generous sponsors, so I would like to thank Numotion Foundation, Biogen, and Genentech for supporting this seminar. I would also like to thank the representatives who are in attendance today. Now I have just a few housekeeping items to go over before we begin our seminar today. We are recording today's event, so we will be posting it to mda.org website for on-demand viewing in a few weeks, and that way we can ensure those who weren't able to join us today can get this information. Please note, for all of you joining us live, all of our phone lines have been muted, and we will be having a Q&A session at the end of each presentation. So utilize the Q&A icon to type in your questions. If you hover over the bottom of your screen, a tray of webinar icons will appear, and if you click on the Q&A icon to open that, and you can enter your question to host. You don't need to wait until the presentations are over either. You can submit questions throughout the entire presentation, so feel free to send those in. And we also encourage participants to communicate with other participants by utilizing the chat feature. And you can just say hi or let participants know where you are from, or if you want to share a bit of your story. You can just click in chat feature and you type into all panelists and attendees, and then you can type away and hopefully meet some people online today as well. You will notice on the agenda there are a few small breaks between some speakers. Please feel free to stay connected to the live broadcast during these times. We will need to set up for the next presenter, so you will hear us on our microphones just to make sure that audio and video is working for the next presenter. But feel free to get something to drink or get a bite to eat. And then finally, we will be sending out a brief survey afterwards, and we would like to receive your feedback on what you heard today. We make sure that we're discussing topics that are of interest to our audience, and so we take your feedback very seriously, and we use that to improve future educational events. So thank you in advance for taking part of that survey. With that, I would like to begin our day and present some information on what MDA is about and what it is we do for the neuromuscular community. We are committed to transforming the lives of people affected by muscular dystrophy, ALS, and other related neuromuscular diseases. We're able to honor this commitment by the work in two areas, through the innovation in care with our 100+ care centers, as well as innovation in science with a focus on research, therapies, and technologies. MDA is an umbrella organization supporting over 43 diseases in the neuromuscular disease space. No other nonprofit supports so many neuromuscular diseases. As an umbrella organization, we have the benefit to leverage key learnings from one disease to inform others. And so to learn more about all of the diseases under our umbrella, you can visit mda.org under the About Neuromuscular Disease heading. The MDA has made a significant investment in advancing the treatments for neuromuscular disease through innovations in science. MDA is the largest source of funding for neuromuscular disease research outside the federal government, $1.4 billion since our inception. MDA's research is directly linked to approved life-changing therapies across multiple neuromuscular diseases. And we've developed a first and only data hub to aggregate healthcare and genetic patient imported data. We've been on the forefront of research since our inception for nearly 70 years, and we are at a time of unprecedented change in the neuromuscular disease space, with more treatments in development than ever. As you can see here, we have invested over $1 billion in research, and we have currently 252 research projects worldwide that we are supporting. On this slide, you can see the progress that has happened in drug discovery over the past 5 years in the neuromuscular space. Treatments are now available for periodic paralysis, DMD, SMA, ALS, Myasthenia Gravis, and LEMS. We are proud to have funded many of these treatments along the development process. MDA's Mover Data Hub is the neuromuscular observational research tool, and this is improving the ability of researchers and healthcare providers to enhance the care and management of individuals living with neuromuscular disease. This is done by driving disease understanding, accelerating therapeutic development, and optimizing health outcomes. Mover collects clinical, genetic, and patient-reported data for multiple diseases to improve the outcomes and to accelerate drug treatment. The diseases that are currently included in our database are limb-girdle, ALS, Becker's, Duchenne, FSHD, Pompe, and SMA. And there are currently 31 care centers across the country participating in Mover, with 60 or more working on steps towards participation. MDA has the largest network of care centers for neuromuscular disease, providing best-in-class comprehensive care. Our care centers ensure a multidisciplinary approach for patient care, which provides patients the opportunity to see multiple clinicians in one visit, allowing comprehensive coordinated care and helping reducing the number of trips that you need to take to the doctor. We have, like I said, over 150 care centers at medical institutions. We have over 2,000 providers working with families at these care centers. And we have tallied over 90,000 annual visits at these care centers. And currently we have over 12,000 individuals participating in clinical trials. You don't have to navigate this journey alone. We are here to help, and our resource center is available to provide one-on-one support via phone or email for individuals and families looking for information about their disease that is covered under our organization. The resource center is staffed by caring professionals, some who are living with neuromuscular disease, and offer a unique perspective to support our community. We also provide information on the care center networks, disease information, community resources, and our Mover program. And the resource staff is available Monday through Friday, 9 to 5 Central Time, and you can reach them, like I said, via phone or email. Another great tool that is available at your fingertips is MDA's Clinical Trial Finder tool. This tool provides a comprehensive list of currently enrolled clinical trials in our neuromuscular disease space. The tool will walk you through some simple questions to direct you to the appropriate trials that meet the criteria you shared. And I will type this address into the chat feature, this MDA/clinical trials, so you can have that for your records as well. With that, I would like to begin our presentation. I would like to introduce to you Dr. Jonathan McKinnon from Las Vegas Clinic. He earned his Doctor of Medicine degree from The Ohio State University of Medicine and Public Health. He then completed his residency in neurology and fellowship in clinical neurophysiology at the Mayo Clinic School of Graduate Medical Education in Arizona. And following this, Dr. McKinnon completed a fellowship in clinical neuromuscular medicine at the University of California, Irvine. He currently is our MDA Care Center Director in Las Vegas. So, Dr. McKinnon, thank you for being with us here today, and I will go ahead and turn the presentation over to you.
Thank you, Nicole. Can you hear me okay?
We can. You can go ahead and share your screen.
Very good. Make sure I've got the right slide here.
Perfect.
Very good. So I appreciate the opportunity that I've been given to present this topic to all of you today. It's in preparing for this lecture, I was given a wide open option of what to present, and knowing that this audience is a fairly diverse group of individuals and family members and support people for muscular dystrophies, I tried to be somewhat all-inclusive in terms of just the breadth of the topic. I hope that I've not bitten off more than I can chew in the process, because there is a whole lot of information to share, and unfortunately that won't allow us to go into great depth into any individual topic. But what I do hope that we'll accomplish today is that in discussing a handful of fairly common diseases that fall under the MDA umbrella, as what in terms of the active clinical trial research that's going on, I hope that I'll be able to share some important information with most of you. With that being said, these are the diseases, the disease conditions that I'm going to be discussing today, and I'm not going to discuss the specifics of the diseases per se, but rather the clinical trials and research that's ongoing in these disease areas, and we'll go through them in this list order, which is ALS, Duchenne muscular dystrophy, SMA, Myasthenia Gravis, inclusion body myositis, limb-girdle muscular dystrophies, as well as Charcot-Marie-Tooth. So you may just glance at this, knowing that any number of you may be more interested in one topic than another. This is the sequence that we're going to be going in. So you can get your sandwich or whatnot, or your breakfast bagel, if the topic order is not a topic that you're presently interested in. So there's a few caveats to this conversation as well, which is that the focus really is on a few different things. One, I am going to discuss recently approved therapies. Part of the goal of clinical trials is to bring drugs to market, and we don't want to forget those drugs, as they've, especially the ones that have recently made to market, having recently gone through the clinical trial process. So I do want to highlight those as we go through the process. We will we'll talk about investigational disease-modifying therapies for each of those disease conditions, primarily those that are intended to slow down the disease or alter the disease. We won't spend quite as much time on the disease, or on the therapies that are intended at treating the symptoms of the disease. And so we will touch to some extent on some of those, but this would certainly not be considered an inclusive list on that regard. And then, of course, phase 3 trials are those trials that are randomized, placebo-controlled trials that are the pivotal studies that bring drugs to market. And we're going to focus on those trials, as those are the drugs that are closest to being potentially approved. Although we will touch on some trials that are phase 2 or perhaps even phase 1, particularly important trials that seem to be offer some major breakthrough potential in the future. So we'll talk about all of these things. I'm not going to discuss specific diagnostic criteria for different disease conditions. I'm not going to specifically discuss mechanisms of action mechanisms of disease, unless it pertains particularly to the clinical trial drug that we're discussing. And as I mentioned, we're not going to discuss symptomatic management of disease per se, although these are clearly very important considerations when you look at all of these diseases. And then my last disclaimer is that this list is by no means to be inclusive. If perhaps I've inadvertently forgotten some therapies that are important or pivotal or critical to this study of the disease or to the understanding of the disease, you'll have to forgive me. There is a lot of information out there in this rapidly expanding area of neuromuscular medicine, and I've done my best to hit the highlights, but definitely it's impossible to hit everything in such a short lecture. So that being the case, let me talk about the way we're going to approach discussing these disease conditions, and a few important points as far as that goes. Genetic diseases have different inheritance patterns, as we all know, and it's important in the understanding of those inheritance patterns in regards to development of a drug that may be effective at treating that disease condition. So I am going to talk to some extent about the genetics of some of these diseases. And specifically, it's important to know that dominantly inherited diseases, as we all know, we each get one copy of our genes from our mother and one copy of our genes from our father. So we have 2 copies at least of every gene in our body. And a dominantly inherited disease means we've inherited a copy from one of our parents that has a toxic gain of function. In other words, you only need one bad copy to cause a disease condition. So that is important in understanding how we would go about treating it, because that gene itself that was inherited from one of our parents is causing problems and it needs to be suppressed or removed. Recessive diseases, alternately, are where there has been 2 bad copies, one from each parent. In other words, 2 bad copies of a gene means that you don't have a normal copy of the gene, and therefore you have lost the normal function of that gene itself. And so for that reason, the treatment approach for recessive diseases would be to restore the lost function, or I should say one treatment approach for a recessive disease would be to restore the lost function of that gene. And then in the case of sex-linked diseases such as Duchenne, but there are others, there is one sex chromosome that comes from the mother and one that comes from the father. Of course, females have 2 X chromosomes and males have 1 X chromosome and 1 Y chromosome. And sex-linked diseases tend to be recessive in the sense that males are more frequently affected because they have one disease copy from the mother, and then the Y chromosome does not have the corresponding gene, and so they're deficient in that gene function. So that is also important in the way that we understand those diseases, partly because we can understand that restoration of that loss of function could potentially restore the function of the individual, or at least improve the function of the individual. And then, of course, genetics, which there will be a lecture on later this morning or this afternoon, is a very complicated thing to understand, and there are many diseases for which there are multiple genes that may be contributory to a disease condition. I'm not going to be discussing that, with the exception of the Charcot-Marie tooth element that at the end of the lecture that some genes are overexpressed, and a means of treating the disease may be to suppress the gene that's overexpressed. So we'll touch on all of these topics, and I just want to make sure that we have a general understanding of those important principles as we go through this process. So that's the genetic disease component. Unfortunately, there are many ways that we can acquire disease, and neuromuscular diseases are no exception. It's not strictly genetics, and there are many other ways of acquiring disease. And the second category here would be the inflammatory or autoimmune diseases, which there can be a dysregulation of the immune system where our bodies are being attacked by our own bodies. And so in those disease conditions, the treatment approach is to suppress the immune system and to stop it from hurting our own bodies. And then there are other disease processes as well, but one that is fairly common is one that we call idiopathic, which just means that we don't really know, or we don't fully understand what's causing a disease. It may not be genetic, it may not be inflammatory, it could be something else, but we don't have a full understanding. And so we use this somewhat blanket term, idiopathic, when we don't really understand. And so some of the diseases that we'll talk about today may fall in that category as well. And strictly speaking, not every disease is just one of these categories. In fact, many diseases, including some of those we'll discuss today, could have components of a genetic process, an inflammatory process, or perhaps another process that's not fully understood. So this is the complexity of neuromuscular diseases. And as we move forward in our understanding of these diseases, we further refine our understanding of which components may be contributing to a particular disease condition. And then, of course, the elephant in the room, which is, you know, we're having this conference as a webinar, partly, well, fully because of this COVID pandemic that is upon us, and it has, in fact, impacted the way we perform clinical trials in this country and across the world. And so I wanted to give a very brief update on how that has impacted research, both for myself, but also for other institutions. Some clinical trials have been delayed as a result of the COVID pandemic. Some centers are not allowing new patients to be recruited or enrolled, or they're restricting recruitment to certain specific clinical trials. Some measures, study metrics, that, as we call them, that we use to monitor the progression of disease have been discontinued. Specifically, respiratory function testing has been diminished to a significant capacity out of concern of risk of exposure to care providers as well as individuals participating. So some trials have had to alter their approach. And so I would consider those three things to be generally negative aspects of the pandemic. Although if we're going to look for a silver lining here, the fourth point is that as a result of this pandemic, remote opportunities or virtual opportunities to participate in clinical trials are increasing. And in some cases, that has expanded access to certain individuals, especially individuals who live distantly from an academic center. So my hope is that as we make our way through this pandemic and ultimately eventually find ourselves on the other side of this pandemic, we'll be in a better place as a result of having to innovate our way through this process. But no doubt this pandemic has caused a slowdown in some respects to clinical research across the country and across the globe. Okay, so all of my prefacing is all finished. Let's jump into the clinical trials for the disease conditions that I mentioned. Briefly, ALS generally is mostly idiopathic. In other words, most people acquire ALS as a result of a sporadic disease process that has no definitive source or explanation. And then this smaller percentage, and I'm estimating 15%, although that number is changing, is hereditary ALS or genetically caused ALS. As we've advanced our understanding of this disease process over the last decade or two, that number of hereditary ALS has gone up from roughly 5% to its present number of roughly 15%. And I would anticipate that if we were to do this lecture 10 years or 20 years from now, that 15% may be higher. But what we definitely understand about ALS is that it probably represents multiple different diseases that have a final common pathway of injury to the motor nerve. And so it's a complicated disease to design a drug or an intervention for, partly because it is, in fact, many different diseases that result in the same final common pathway. And so there are, as you may understand or as you may imagine, that would allow for a number of different approaches to trying to treat or slow the disease progression in ALS. As of today, there are currently 2 approved therapies for ALS. One of them has been around for a good quarter of a century, which is Rilotec or Riluzole, and it does have a small benefit in terms of the overall survival in ALS of 3 months. It blocks the excitatory neurotransmission of glutamate, and it does, in fact, slow the disease progression, but marginally. More recently, a new drug came around in 2017, Radicava, also known as edaravone. It has antioxidant properties, and it also slows the progression of ALS. It wasn't measured in the same way that Rilotec was measured because it doesn't, it was studied in early disease and not late disease, and therefore it's hard to estimate exactly what the survival benefit might be, but it's believed to be better or more efficacious than Riluzole, although an absolute survival benefit can't be determined. We'll probably get more data in the future as far as what that actually means in terms of survival benefit. It's a little more cumbersome. Riluzole is a pill, you take it twice a day. Radicava is an infusion, and it's 2 weeks of the month is for an hour-long infusion. So in many cases, because of the frequency of the infusion, you have to have a port placed, and it's a bit more cumbersome to administer. But it nonetheless is likely to be a more effective drug at slowing down ALS. And for that reason, we're very happy that it has come about and is making its way into the clinical practice. When I first put together a topic of clinical trials for ALS that was in my fellowship in 2011, and there were these four study drugs being considered. And we're going to talk about some of these in more detail in a few minutes, but the reason why I included this slide is that there were four treatments being studied for clinical trial purposes in 2011. As of this year, in 2020, we are significantly further along in terms of the number of treatments that are being looked at in terms of slowing or stopping the progression in ALS. So we're definitely further ahead in terms of numbers of therapies being looked at, and 2020 stands to be a breakthrough year as far as clinical trials are concerned in terms of the way we advance our understanding for ALS management. These categories of disease or of mechanisms, I should say, I'm going to go through each of these categories briefly to talk about different therapies that are being considered. Some of these are in phase 2 clinical trials, some are in phase 3 or soon to be in phase 3 clinical trials. And of course, all of these were set to get going this year, and I can say that for the majority of them, the brakes have been pressed in terms of getting these up and running. But some of them are up and running and are available as clinical trials even as we speak. So just briefly going through these categories, anti-inflammatory disease modification is being looked at in terms of complement inhibition. Both of these drugs, ravulizumab and zilucoplan, are C5 complement inhibitors. It's a complex mechanism of complement is important in cell destruction, especially when it comes to pathogens or infections invading our bodies. The complement system is important in stopping infections. But as it turns out, it's also important in regulating a number of disease processes, and there's reasonable data to support that complement may be playing a role in the loss of motor neurons in ALS. And so for that reason, these two drugs are being looked at. The ravulizumab is a monoclonal antibody, as zilucoplan is a small molecule, and they both have clinical trials that are starting for ALS. Antioxidants, we know that oxidative stress is harmful to the motor nerves, and so far radicava and riluzole seem to have some antioxidant properties, and there are other medications, and the terminology is going to get a little confusing here, because when things are studied as a clinical trial, they don't always have easy to say or easy to pronounce names. So I'll do my best to describe all of these, but realize that if any of these treatments do come to market, they may come to market eventually with a different name. So it's probably not so critical that you commit to memory the exact description of the molecule, but it's important to understand, I believe, the mechanisms that are being considered. So this copper ATSM is based on the SOD1, which is an inherited form of ALS, where there is a loss of copper-containing SOD1, and so this molecule is being considered to treat the disease by adding extra copper, and thereby improving oxidative stress. Verdiperstat is an antioxidant, and it does cross the blood-brain barrier. And then CNMAU8, which is a gold nanoparticle, can serve as an electron reservoir, all of which are believed, all of these mechanisms may be beneficial in relieving the oxidative stress that occurs in the case of ALS. Some additional anti-inflammatory medications that we're looking at are masitinib, which is a tyrosine kinase inhibitor. It blocks mast cells and macrophages, which are cells that are part of the immune system that help that in the normal condition would be releasing their toxic substances on an infectious pathogen, but in the case of ALS may be causing damage to the nervous system. Ibutylast is a small molecule that has a role in inhibiting phosphodiesterase 4 and 10, and those are known to be macrophage migration inhibitors. So, and then Tecfidera is the drug that's already approved for multiple sclerosis, and it's important in the immune regulation of trafficking of lymphocytes. And there's some reason to believe that there may be a role of lymphocyte trafficking in ALS as well. Additional anti-inflammatory drugs that are being considered: Actemra, which is a commercially available drug indicated for rheumatoid arthritis and other autoimmune diseases, is an interleukin-6 inhibitor. It has a direct impact on the inflammatory response and can reduce the marker, which is C-reactive protein, which is a marker of inflammation in the body. We know that in some patients with ALS, particularly those with rapidly progressive ALS, C-reactive protein is elevated. So we're using circumstantial evidence to some extent to understand what things may be effective, and then ultimately the studies have to be done in order to determine if these treatments will be effective. And then Treg and IL-2, these are regulatory T lymphocytes, which are removed from the individual, expanded outside of the body, and then reinfused, and they may be effective at reducing inflammatory response in the individual as well. So that is being considered. There are a number, there's another novel molecule being considered. It's RNS60. It's an oxygen surrounded by a water nanobubble, and it is believed to have some anti-inflammatory properties, as well as activates antioxidant pathways. There are a couple of medications that the mechanism is intended to alter the axon excitability. In other words, to reduce the threshold for firing. Retigabine is a seizure medicine. It's a potassium channel blocker, and it may be effective at slowing the strength duration time constant, which may have protective effects for the motor neuron. Mexiletine is an antiarrhythmic medicine that's used by cardiologists in certain instances. It's a sodium channel blocker, and it may be effective at relieving some symptomatic cramping in ALS. And then there are apoptotic, which is the word that we use when we're talking about a cell dying. And so these medications are intended to protect the cell from dying when it's under stress. So arimoclomol, it increases the production of heat shock protein 70, and that may be important in clearance of certain proteins that can form within the cell of the motor nerve. AMX 0035, it is also important in oxidative stress of the mitochondria, and it may be effective at reducing the cell death from hydrogen peroxide. And then there are some additional neuroprotective mechanisms, medications being considered, which may be important in the regulation of the cell death apoptosis activating system, and or upregulating, in case of the pridopidine, is important in the upregulation of brain-derived natriuretic peptide, which is important in calcium signaling and cell survival. So it's complicated, the specifics in terms of how these drugs may be effective, but ultimately there are a number of different ways of trying to get at this disease condition and trying to protect those motor nerves. I won't go into great detail in these two, but these are meant more to improve the function in ALS regarding to the skeletal muscles and the rate of progression of decline of the skeletal muscles and the overall function of the skeletal muscles as they decline. Retrovirals have been looked at and are being considered. They may play a role as far as modulating the inflammatory response, and their phase 2 data is showing some promise as slowing the rate of progression as well. And of course, we couldn't talk about ALS therapeutics without talking about stem cell therapies. There are, of course, these clinical trials have been going on for well over a decade now. And there are some promising results in the context of some of these clinical trials. We're still we're still awaiting the final results of these stem cell therapy trials, and I'm hopeful that the results will be encouraging, particularly given the number of years and research dollars that have gone into stem cell therapies for ALS. And to some extent, the belief is that stem cell therapy may be helpful not at necessarily restoring the lost motor nerves, but at restoring the balance of the extracellular or the fluids around those motor nerves that are under stress, and may help to support those motor nerves from degenerating and keep them from degenerating. Although the mechanisms are likely very diverse in stem cell therapy as well. And then lastly is on the topic of ALS. I know we've spent quite a bit of time here talking about ALS thus far, but there is a new approach to conducting clinical trials for ALS, and this was being piloted by the Niels Group, which is the New England ALS group, and this approach is to include more than one study drug for individuals engaged in a clinical trial. And the reason for that is that every clinical trial has a placebo arm. Typically, it would be 1-to-1. One group, 50% of people would get the drug and 50% would get a placebo, or in some cases 2-to-1, where 66% of people will get the drug and 33% will not get the drug. Well, as you can imagine, most people are not interested in getting a placebo, this platform drug trial concept is that you can include the placebo arms from multiple different drug arms and use them as the control group to compare to, and thereby increasing the number of people who actually get the study drug, so that instead of 50% or 60+% getting the actual drug, you may end up with 80 or 90% of individuals participating in the study who actually are able to get the study drug. So these are, it's a very exciting approach. It has tremendous potential benefit for the patient as well as for the research process, as well as it has the potential to study more drugs in a shorter period of time and ensure that more people are exposed to actual drugs as opposed to placebos. So I'm going to switch gears here now, and we're going to move to Duchenne, and we're going to talk about treatments that are coming down the pipeline for Duchenne. As we discussed earlier, it's an X-linked recessive disease, which means that the gene that is responsible for the disease process is called the dystrophin gene, and it is a very large gene, and the gene itself, when it's functioning properly, creates a very large dystrophin protein. As it turns out, that protein is essential to making sure that the muscle fibers within the cell are linked properly to the extracellular components that ultimately link up with the tendons. And so in the case of Duchenne, when you lose this large key protein, you have a dislinked muscle fiber with a dislinked with the tendons, and the muscle doesn't form properly, and ultimately over time the muscle degrades. It results in progressive muscle weakness. It's important to mention Becker in this regard because Becker muscular dystrophy, like Duchenne, is a mutation of the dystrophin gene, which results in a dysfunctional dystrophin protein, but a less severe dysfunction of the protein. So a partially effective gene and a partially effective protein. And we're going to see in a few minutes where that process was key in helping us understand how to develop a potential treatment for Duchenne. As of today, these are the treatments that are approved in the United States for Duchenne muscular dystrophy. We know that Duchenne does have an there is inflammation in Duchenne, and suppressing the inflammatory process does help to slow down the disease. So in that regard, prednisone and Emflaza, also known as deflazacort, are used as anti-inflammatories, and they are effective at slowing down the disease. There are also genetic-based therapies, that two in particular, that are presently approved. Exondys 51, also known as eteplirsen, and Vyondys 53, also known as golodirsen, which were recently approved in the last couple of years for specifically targeting individuals with specific mutations of the dystrophin gene. And it's clever design because the dystrophin gene results in 60+ coded proteins that are eventually assembled together to make the final fully functioning dystrophin protein. And knowing that Becker is is a mutation where there's a partially functioning gene and a partially functioning protein. By genetic design, we have been able to link the functioning portions of those proteins together to create a partially effective dystrophin protein that has a partial efficacy. And so this is termed or coined exon skipping, because you're skipping the bad components of the gene and you're linking together the effectively good components of the gene. And it has been shown to slow down the disease process to a certain extent. So there are a number of therapies that are exon skipping therapies that are moving through the pipeline. We're going to talk about some of those in a second here. But as of today, there are 2 that are currently FDA approved. And I should say that it's important that we understand that everyone who has Duchenne muscular dystrophy be tested and have a defined genetic mutation in order to determine who is going to be a candidate for one of these therapies or potential other therapies in the future. And then the last part of this slide is that gene replacement therapies, as of today, there are none. But we're going to talk about that in just a second as well. So what are the clinical trials? Well, in terms of immunomodulatory or inflammatory modulation, there are several drugs that are being considered as candidate drugs for Duchenne. These are currently in phase 3 or phase 2 clinical trials. Etiselenexant is a selectively immunomodulatory medication, which selectively has we believe selectively inhibits the inflammatory response, and it may have some beneficial, it's hoped to have some beneficial effect at slowing down Duchenne. Tamoxifen is an estrogen receptor modulator. Vamorolone is a steroid alternative, and then pamrevlumab is a monoclonal antibody to connective tissue growth factor, which may limit the formation of fibrosis. So all of these may be or are being looked at and may ultimately, some of them hopefully ultimately will be deemed effective at slowing down the inflammatory aspect of Duchenne. I mentioned the exon skipping therapies that are approved. Exon 45 amenable, there's a medication, a drug, casimersen, which has the phase 3 trial has completed, and the FDA is reviewing this to determine if it meets key thresholds for approval. And again, it's specifically to exon 45 amenable mutations. Translarna, also known as Alurin, is a medication that suppresses this a nonsense mutation or an abnormal stop codon, as we call it in the genetics world, to allow the gene to be continued to transcribed and allow the full protein to be created, as opposed to just a partial protein. So, and again, that's very specific to certain mutations within the dystrophin gene, but may ultimately be effective at creating some functional dystrophin for certain individuals. And then viltelarsen is an exon 53 amenable therapy that is working its way through the pipeline as well. We're going to spend probably a few minutes on this slide, but just because of the relative importance of these 2 therapies that are being developed, one by Pfizer, the other by Sarepta. These are two genes that are being incorporated into a virus that then ultimately will hopefully find its way into the human cells. So as you can imagine, when you have a genetic disease where there is an absence of the necessary gene to create the necessary protein, it creates a very challenging scientific dilemma of how to effectively treat that disease. In the case of Duchenne, where there's a very large gene, the dystrophin gene, it creates an additional challenge of being able to get that gene into all the cells of the body. But over the course of at least 2 decades, this novel treatment approach has been developed, wherein the gene can be effectively included into a viral vector, and then the virus, or the viral vector, I should say, would be infused into the individual, and then, as viruses do, they find their ways into cells and deliver the desired gene product into the muscles of the body. These are clearly modified viruses. These are not the actual live viruses that are found in the real world, but we're using their viral technology, so to speak, to deliver the necessary gene into the cell. Because the dystrophin gene is so large, this virus is not capable of including the entire gene. And so, as I mentioned earlier, as is similar to the case of Becker muscular dystrophy, there have been found mutations within the dystrophin gene that have essentially normal function, but they are much smaller than the full dystrophin gene. And so we're using those forms of the dystrophin gene to include into a viral vector that then ultimately can be infused into an individual, and with the hope of restoring or improving muscle function. And so far, these studies are ongoing. The data has not been published. They are not completed. But we do have hope, and as we're going to talk in a few minutes, from the SMA clinical trials, it has been shown to be effective in the case of SMA. And so we have high hopes for one or both of these treatments to be effective. It's not without risks. There are this AAV virus has some known potential toxicity to the liver in the case of SMA, and so it has some challenges, but nonetheless, we do have high hopes that one or both of these may be effective. And in the case of Duchenne, where if it can be identified early in life, it's hoped, at least by me, that the destructive processes that affect the muscles would be significantly slowed or perhaps stopped with restoration of the normal gene into the muscle fiber. So it has great potential, and we're looking forward to these treatments as they make their way through phase 3 trials. I am going to move on to SMA now, and I hope I'm not, I know there's a lot of information in these slides, and I hope I'm not overwhelming anyone here, but I think that there are themes that probably carry over from one disease condition to another that I hope you'll find useful, even if you're listening for only one particular disease condition. So in the case of SMA, as I mentioned earlier, it is an autosomal recessive disease. In other words, no normal copies of the gene in the individual. It's a loss of the SMN1 gene, and it results in the motor nerve degenerating. It's frequently an infantile or childhood onset disease, although there are adult onset forms. As of today, there are 2 approved therapies for SMA. Spinraza, also known as nusinersen, upregulates the SMN2 gene. And SMN2 is essentially a backup copy that is not as good as the SMN1 gene, and we have several, most people have several SMN2 genes that can be upregulated, and they can be partially functioning in a way that can help slow down the disease progression. And as the pivotal study showed in Spinraza, it does slow down the disease progression when you can increase the SMN2 copies. And then, of course, Zolgensma was recently approved, which is in fact the gene therapy replacement for the SMN1 gene. It does use the AAV9 as the viral vector, and it was approved last year for ages up to 24 months. It was studied in kids under the age of 6 months, and so the approval was for an expanded age of the individuals. And as this drug has shown, it has had, in many cases, profound benefit to individuals who were given this treatment. Instead of regressing or losing motor milestones, many of these individuals have gained milestones. And so that has been an incredibly powerful new therapy to market, and we're very excited that it has made it to market. As I mentioned, it's a recessive disease. I think I'm going the wrong way here. So gene-based therapies, there are ways, other molecules that are being looked at that may also upregulate the SMN2 gene, as is the case in risdiplam. That pivotal study has been completed, and the FDA is reviewing that. It is an oral therapy as opposed to an infusion, which is the case for the Spinraza. And then branaplam also is being looked at as a SMN2 upregulator as an oral administration. And then Avexis as a somewhat of a piggyback of the Zolgensma therapy, they're looking at expanding this the patient age range from up to 2 years at the present up to a greater age range of up to 5 years. And so this current clinical trial of the 101IT is intended to study this drug in an older age range and ultimately, hopefully, determine if it works for the older kids as well. I know I'm not running out of time, but I'm getting closer to running out of time. So I'm going to go a little quicker now with a few other disease conditions, and then we'll allow some time for questions at the end. So myasthenia gravis is an autoimmune disease. It's an attack of the motor and of the neuromuscular junction where the nerves speak to the muscles. It can be, people with myasthenia have fluctuating weakness, especially eye muscles and respiratory muscles. And it is an antibody-mediated disease, and it's responsive to immunomodulatory therapy. As of today, these are the treatments that are indicated for myasthenia gravis, including prednisone, Imuran, Cellcept. They're not officially indicated, but frequently used. IVIG, plasmapheresis, typically in a crisis situation, although they can be used for some individuals in a chronic fashion. And then a few years back, eculizumab, also known as Soliris, was FDA approved for treatment of myasthenia. And as I mentioned earlier, I didn't mention earlier, this drug has its a mechanism of action at the complement inhibition, also a C5 complement inhibitor, regulating the inflammatory response. There are several clinical trials going on for myasthenia gravis. Ravulizumab is a cousin molecule to eculizumab. They have modified the molecule to allow for an extended half-life within the bloodstream, and it has its efficacy so that it requires only every 8 weeks an infusion instead of every 2 weeks, which is the case for eculizumab. So Leucoplan, which I mentioned earlier, is being also studied for ALS, is the daily subcutaneous injection, but it's a small molecule complement inhibitor. And rozanolixizumab is an FC receptor antibody, which is being looked at and is as a weekly subcutaneous infusion. And its role is that it removes circulating IgG antibodies and by inhibiting the recycling of those antibodies. And so it may also prove to be effective in regulating myasthenia by removing pathogenic antibodies. Inclusion body myositis, we don't know ultimately what is causing this disease. We do know that there are inclusions that are found within the muscles themselves, and that ultimately the muscles become weak. Prominently, the forearm flexors and the quadriceps muscles are frequently involved. As of today, there are no approved therapies for inclusion body myositis, in spite of it being a fairly common neuromuscular disease. As I mentioned earlier, arimoclomol has effects on the heat shock protein and may result in decreased protein clustering within the cells. And the clinical trial has been completed. We're hoping to see some results in 2021. And then I included just the last couple of slides here, discussion about limb-girdle muscular dystrophy, partly because it's a fairly common neuromuscular disease, although it really is many different diseases that result in the limb-girdle muscles becoming weak. And so ultimately, these are many different diseases that have different treatment approaches. And as is the case, there are dominantly inherited forms, recessively inherited forms, as well as X-links, et cetera. And we're particularly optimistic about the recessively inherited diseases, partly because of the potential for viral vector-related gene therapies, which are coming down the pipeline for these particular diseases as well. And then Charcot-Marie-Tooth, similar to the spectrum of limb-girdle muscular dystrophies, is a number of different diseases that do ultimately cause the final common pathway of a neurodegeneration and distal weakness. They can be inherited in a number of different ways, and they are being considered, treatment approaches are being considered for a number of these. One of these would be the PXT-3003 study, which is a combination therapy of baclofen, naltrexone, and sorbitol, which are commercially available medications, with the hope that it will target PMP22 gene expression, because CMT, the most common form of CMT is CMT1A, and the cause of that is a duplication or an extra copy of the PMP22 gene. And so the goal here with this particular treatment is to suppress the PMP22 gene expression and restore the cell back to a normal PMP22 expression, and thereby hopefully reduce the toxic effect on the nerves themselves. So I just include this slide partly because I don't have any other visuals in this presentation. I always like to have at least something different than words to look at. But I think we can look at this as the current landscape of clinical trials in neuromuscular diseases as either a half full glass or a half empty glass. Clearly, we have room to progress and to do better, but there's reasons to be optimistic in that there are a number of ongoing clinical trials that have shown some degree of promise and hope for the future. And personally, I'm hopeful that this is, as we move forward into the next decade in particular, we will see some major advances in the management of these diseases. We have a large mountain to climb. This is Mount McKinley or Denali in Alaska, but our goal is to get to the top, and I do hope that eventually we'll have significant improvement in management of these diseases. I'm going to stop there, and I know there may be some questions that have come up over the course of this lecture, so I'm going to go ahead and Nicole, I see you there, so maybe you want to be the one to read through the questions.
Sure. Thank you very much. Well, we do have a couple that came in. One of them is around the FDA. Do you know now that COVID has hit us, what is the expected time frame, I guess pre-COVID and now during COVID, that FDA would take to approve a new drug?
Well, I don't have any inside information, so I'll be upfront about that. Mm-hmm. What I can say is that I would hope and I would expect that that the approval process itself would not necessarily be extended, significantly, probably somewhat, just based on the fact that everyone is having to modify their ways that they work, and it's possible that individuals at the FDA are now doing remote conferences and so forth, just as are we. But I would not expect that would result in a major delay in the overall review of data and those types of things. To me, the more likely source of delay in getting drugs to market would just be the clinical trials themselves, and the more cumbersome nature of of just making sure that everyone who's involved is participating in a safe way. As I mentioned, probably the biggest slowdown is on the level of the institutions, where there have been, in some cases, everything has gone to remote visits with physicians, and you can't, in most cases, clinical trials are hard to administer remotely. So just the enrollment process itself has taken a huge slowdown. But ultimately, we do hope that those slowdowns are gradually eased and that the trials will continue to move forward, those that are on pause or on hold, and that, yes, eventually the data will make its way to the FDA and ultimately, hopefully, to market.
Mm-hmm. Any recent developments for Becker treatment?
So that's a good question. Becker is interesting, partly because we really don't have good hard data to guide treatment decisions. And in many cases, the new therapies that are being developed for Duchenne are really strictly indicated for Duchenne muscular dystrophy and not Becker. So I guess the short answer to this is that no, there aren't any new treatments for Becker that I am aware of, other than to say that the immunomodulatory therapies that we use for Duchenne, which include prednisone or deflazacort, in some cases can be used. And it's possible that as some of these other immunomodulatory therapies come along, perhaps they will make their way into use for Becker as well. There aren't as many individuals with Becker, which is in some ways a good thing. And for that reason, it's hard to do clinical trials for individuals with Becker. But I do think that at some point, those trials probably will happen. And it's possible that potentially some of the exon skipping therapies, in the case where it's a Becker individual with an exon that has an existing exon skipping therapy, I would hope, and again, I have no inside information that this would ever necessarily be the case, but I would hope that potentially some of those individuals may be eligible for exon skipping therapies as well.
And someone chatted in, could DMD successful trials or treatment be applied to the future to BMD? So that is the goal then, correct, since there are so few Becker?
Yeah, so that I think that's probably a succinct way of saying what I think I just said, which is, and we see that in other disease conditions as well. For instance, in the ALS world, we get a drug approved for ALS, but then there are other similar diseases to ALS like progressive bulbar palsy or primary muscular atrophy or other things like that, that those therapies may ultimately work for those conditions as well. So yes, I think we do see that. It's tricky. I can't speak with any degree of certainty. These are very expensive therapies, and you want to make sure that you're not just on the matter of expense, but on the matter of safety, that you're using them for appropriate individuals. But I think in the case of Duchenne and Becker, you could make an argument that the safety profile would not be significantly different for a Becker individual than for Duchenne.
Okay. Are you familiar with vitamin C helping CMT?
So there are a number of different vitamins that work or that are used to treat different neuromuscular diseases, including CMT. And it's a topic that comes up quite frequently. To my knowledge, no specific vitamin therapy has ever been established to be effective at slowing down any neuromuscular condition. which includes CMT, other than to say, but with the caveat that I'm not sure that we do a great job of large randomized controlled trials as far as vitamin therapies are concerned either. So my usual suggestion, and again, this varies on the individual, but we try not to do high doses when we're not sure what the effects might be, but standard doses of vitamins in a condition where there's a nerve or a muscle disease potentially could be helpful and hopefully not harmful. And so my usual suggestion is to stick to standard dosing of vitamin therapies if you choose to use vitamin therapies, although there's no hard data to show that it actually slows down the disease.
Okay, great. Well, I do not see any other questions in the queue. So thank you very much for your presentation and being with us this morning. I know it's kind of early on your side, so I hope you enjoy the rest of your day. Thank you so much.
Thank you.
All right, and up next, we will be hearing from Stacy Garrison, who is our registered dietitian, and we will be discussing nutrition and neuromuscular disease at 10:20. So feel free to take a break, and we will be back at that time. Thank you.